Hi there, and welcome to Smith and Sheridan, a podcast on the science and business of biotechnology, presented by me, Cormac Sheridan.
And me, Andy Smith. Hello, Cormac. Hello, everyone. How are you this week, Cormac?
I'm good, Andy, and I'm feeling a bit more optimistic about the area that we're going to discuss this week than I had been before I started doing some research. We're looking, folks, this week at the broad area of brain cancer, primary malignant tumours of the brain, the ones that are aggressive and or can become aggressive and generally lead to a very grim prognosis and outcome. And it's been just an area that's been very difficult to treat, but there have been some signs to give us grounds for optimism, tentative optimism, but there is some growing indications that the successes that we've seen in other cancers using immunotherapy approaches can be applied to brain cancers, and in some instances, maybe at the level of a case report rather than clinical trials. We're starting to see possibilities emerge, and that also includes, say, CAR-Ts as well as immune checkpoint inhibitors. All early stage stuff. Last year, there was a much hyped clinical trial report which was worthy of the hype. I'm referring to a drug that was originally discovered by Agios Pharma but picked up by Servier. They have a small molecule drug called vorasidenib, which is a dual inhibitor of two mutated isoforms of isocitrate dehydrogenase. Those mutations play an important role in certain gliomas. We can get onto the details of it later, but that was hailed as the sort of first real breakthrough in the treatment of brain cancer in about 2 decades. And it's currently under regulatory review, both in Europe and in the US, under the FDA's PDUFA rules. The action date is the 20th of August. And I would be, and everybody else would be gobsmacked, obviously, if that didn't get an approval based on what they've achieved. Basically, they got the more than doubled progression-free survival in post-operative patients with recurrent or residual IDH mutant gliomas. So the median PFS for those on the drug was 27.7 months versus 11.1 months for those who only got supportive care. I know that you were interested in this, was piqued as well by the extraordinary story coming out of Australia.
Yeah.
Professor Richard Scolyer, himself a very distinguished pathologist and researcher in the area of melanoma, he was diagnosed with glioblastoma at stage 4 because he basically experienced seizures. And he has been on this extraordinary cocktail of immunotherapy drugs, personalized mRNA vaccine, as well as undergoing surgery and radiotherapy. And more than 1 year out from diagnosis and treatment, he is still recurrence-free, which is an extraordinarily encouraging outcome. It's gotten a lot of attention. What's fascinating to me is that his long-time research colleague in the area of melanoma, Georgina Long, who has also led the work on his treatment, she has published, still available only as preprint, but as sort of a case report detailing the progress so far in his therapy. Absolutely extraordinary stuff. So there's other things going on too, but they, I guess, are the recently 2 different CAR-T therapies went into patients as well, which was also fascinating.
I have to say, Cormac, this is the episode that I never really wanted to record on brain cancer, just because for many years as an investor, it was a wasteland or a desert of new therapies. I mean, the 20 years, the horrible triple chemotherapies, and even gemcitabine, they're not nice drugs. Sure, they're nicer than dying of brain cancer, but there were...
And they prolong the inevitable, they're not going to cure, typically.
Yeah, which is the horrible thing, right? I mean, you're a brain cancer patient, you have a bit of your brain cut out as part of surgery, you might have radiation therapy, you have these horrible chemotherapies, and then all you can look forward to is what oncologists call remission, but are careful not to call a cure. And that's for many cancers. Brain cancer seems to be one of those to which you eventually succumb. As an investor, I would see companies developing drugs, and it is often for glioblastoma or malignant gliomas, just because that is the most commonly diagnosed brain cancers. But there are many others. I hesitate to say hundreds, but depending on which tissues, which types of the brain, it could be virtually anywhere.
It's easily over 120 or 150. Yeah. I'm not sure the exact number myself, but it's definitely over 100.
And depending where they are in the brain, they're probably inoperable or very difficult to operate on.
Yeah.
And this is partly why I never wanted to record this episode. I'm sure listeners can detect that we're not laughing and joking about this. This is a sobering episode today, I'm afraid. So if you're looking for laughs, this was not the one. And more recently, as you mentioned, Cormac, we saw the single N=1 case study of this professor in Australia, but also my son's friend, unfortunately and very sadly succumbed to glioblastoma multiforme in his 20s. And I'd like this episode to be dedicated to Jacob Whitehead's memory. But I mean, to get brain cancer, sure, it's unlucky. But for glioblastoma multiforme, where the average age of incidence is between 40 and 70, and you're in your 20s, I mean, that's horrible. Yeah. So it was until recently, and you've shown me data that we've looked at the drugs that have been approved, the MEK inhibitors, the BRAF inhibitors, but it's only in the last 2 years that this glimmer of light of new treatments, and even in the last year, have actually come to fruition. Yeah, and there is talk of CAR-Ts, there is a talk of other immunotherapies, and I hope they come quick enough. As you say, I hope the regulators appreciate that there has been very little out there for these patients.
I think there's no doubt that the regulators know it as well. It's usually the sort of blunderbuss approach with chemotherapy, it's radiotherapy, and it's surgery. And also, I mean, I think the new agents, people aren't really talking about cures as yet by any amount, but they are talking maybe about additional life free from complications. And an extra year could be very meaningful for somebody who's facing the inevitable. And that's kind of the sort of commentary, say, that was surrounding the progress last year with the Serbia product vorasidenib. Maybe we should talk about that first because it is close to approval. It's an August 20 PDUFA date, and this is a drug that should be shipping, at least in the US, before the end of the year. And in Europe, with the usual delays around the formalization of the approval and reimbursement, it might take a little bit longer, but it surely should become available in Europe next year as well, because, as you stated so eloquently, the need is huge. There's really, really impressive science, both medicinal chemistry and clinical science and biological research has gone into this. What's really interesting is that isocitrate dehydrogenase is an enzyme of the citric acid cycle. And it ordinarily, the wild-type enzyme, and it's not affected by mutations, it catalyzes the oxidative decarboxylation of isocitrate to alpha-ketoglutarate and carbon dioxide. But when either isoform of the enzyme, IDH1 or IDH2, is mutated, you actually get instead of the alpha-ketoglutarate metabolite, you get what's called 2-hydroxyglutarate, which people call an oncometabolite. It in itself drives cancerous transformation because it accumulates in the tissues, it's not broken down, and it competitively inhibits other enzymes that act on alpha-ketoglutarate, because obviously structurally resembles it. And it results in changes in DNA hydroxymethylation, gene expression, cellular differentiation, and the tumor microenvironment itself. So in itself, it's a driver, which is quite an extraordinary biological insight to get.
And it's strange, isn't it? I mean, when you think the glutamate or glutamate metabolites are important for signaling and messaging in the brain, amongst other tissues, but where you have genes encoding proteins like IDH that have to work particularly hard, and in this case, it's a gene encoding IDH in the brain. In testicular cancer, it's going to be there. There's turning over, people in sunny environments where they get melanoma, where we get tissues that work exceptionally hard, the turnover of those tissues can result in cancer. And you'd think the brain is an isolated place. It certainly is immunologically. I mean, this is why we've not had immunotherapies for brain, just because the drugs have got to get access, got to get past the blood-brain barrier. And that doesn't happen very often. It happens if you get an infection in the brain. Yeah. So as well as finding the right targets and getting the right drugs, you've got to get them to the site of action, which in the brain is generally isolated. And
that's to the great credit of Agios Pharma, the people there who discovered the drug. There's already 2 other ones out there, which I think both originated in Agios originally, but they got picked up by other companies. Ivosidenib and inasidenib. One's approved for AML and a form of bile duct cancer. The other is just for AML only, acute myeloid leukemia, but they weren't brain penetrant. And therefore, the whole design of vorasidenib, it was explicitly seeking a brain penetrant molecule.
Hats off to those chemists and those biologists who went looking for a blood-brain penetrant IDH inhibitor, because there are other bigger cancers you design a program for, but that they've actually gone for this indication.
Yeah, and without going too deeply into the medicinal chemistry, lower polarity and fewer hydrogen bond donors and acceptors, it just means it's less reactive as a molecule as it passes through the membrane. That's a simplistic take on it, but it does result, certainly in mice, they saw dramatic reductions in the activity of the enzymes, the mutant enzymes. So it does work. And then we're seeing it really works in patients. And
which is not typical of neurology, is it? I mean, you tend to think of neurological diseases and you measure mice and their ability to walk through mazes. And I've said it before, I tend to think that us humans, our brains are a bit more complicated than mice. So whatever drug development intervention typically works in mice and is measured by a mouse walking through a maze or a water thing, yet not necessarily doesn't work in humans.
Yeah, but I mean, don't forget that even though the anatomical site of brain cancer is the central nervous system, it affects the central nervous system, but it's not a disease of central nervous system functioning. So there is a certain difference in there.
But in this case, and in any event, I'm happy that vorasidenib works in both.
Yeah, and the biggest safety signal was sort of liver tox in about 10% of patients, and that generally is kind of manageable.
And reversible, typically,
Yeah. So I'm assuming that this is going to be a backbone therapy. And obviously, it's in glioma where the testing and the regulatory review has happened, but they will try it in glioblastoma as well, because basically that is purely just a more advanced form of glioma. And it's typically glioma when it reaches stage 4, when it's aggressive and invasive. So...
But this is a possible downside. I mean, sure, for those patients with the right IDH gene mutation for their glioma, this is going to improve their prognosis no end. And we're at this paradox of gene-directed therapies or drugs directed to mutations in certain cancers is not the same in every cancer. So there are plenty of patients who won't qualify for this drug just because they have a different mutation for their brain cancer.
Well, the good news is, though, is that the IDH mutations are extremely common in glioma. So the majority of patients, I think, will be able to benefit from this.
And glioma is the most common brain cancer, so that's a positive.
Yeah, it is. It's going to become a backbone therapy, which one will hope will help a lot of people. But then maybe that's an interesting point to look at it in a bit more detail at Professor Richard Scollier, because he actually was isocitrate dehydrogenase wild type. And so he wasn't put on this experimental drug. And obviously the kind of career that he's a highly networked and very successful medical scientist. And I think he basically made his illness a research opportunity for his colleagues, and he allowed himself to be a guinea pig. And what they've essentially done is apply a protocol that has had some success in melanoma to the treatment of his glioblastoma. And what they're particularly anxious to emphasize is the necessity of giving combination immune checkpoint inhibitor therapy in newly diagnosed patients before surgery. Because there were sort of half-hearted efforts in the past. They mentioned themselves the Checkmate 143 study, which was comparing nivolumab, the PD-1 inhibitor, with bevacizumab, which is a long-standing angiogenic therapy.
Anti-VEGF,
Yes, and that's long been a backbone therapy, which provides some PFS kind of benefit. And essentially, nivolumab and bevacizumab, the median overall survival was, 9.8 months versus 10 months. 12-month overall survival was the same, 42%. So there was no huge difference. Funny enough, though, the objective response rate was much higher in the bevacizumab arm than in the nivolumab arm. But leaving all that aside, that the bevacizumab original approval was in 2009. And nivolumab, the study was obviously done. It was published in 21st of May in JAMA Oncology, 21st of May 2020. If anybody wants to get the reference, that's the online publication date. So, it didn't move the needle. It didn't kind of add a whole lot. But the point being is that Scolier's team basically were advocating the fact that you need more than one agent and you need to give it before therapy. And what's also really important is that you don't give either prior or concomitant immunosuppressive therapy, which is kind of a no-brainer. But the idea that you still give corticosteroids or you still give chemotherapy when it's a whole new ballgame, you're trying to de-repress
when you want an immune response against the target.
Yes. So what they've done is that he got 12 days before surgery, he got nivolumab, the PD-1 inhibitor, and he got relatlimab, which is a LAG-3 inhibitor, which is only, as far as I know, approved with nivolumab for treating, I think, ovarian cancer. It doesn't have single agent approval yet. He also received ipilimumab, the CTLA-4 inhibitor, the first checkpoint inhibitor to get approval. So pre and post therapy biopsies showed a tenfold increase in the numbers of T cells, and CD3 positive, CD3 and CD4 positive, and CD3 and CD8 positive, tenfold. So they basically are claiming that it's reshaped the sort of immune landscape of the tumour. And basically that's why he appears to be in remission.
But I'm sure the clinical operations people in pharma companies would hate this one case study because it is a mosaic or a hodgepodge, if you like, or a mix-max of things that have been tried before and not worked, and then things that are still experimental, as you suggested, like the mRNA vaccine.
And he's also getting that, by the way, too. I forgot to mention, because in the preprint that Georgina Long has published with her colleagues, they've mentioned the fact that he's getting a peptide vaccine course, which is ongoing. As far as we both understand it, it's an mRNA vaccine that obviously encodes some peptides that are...
But those peptides are tailored to the patient's particular tumour. So from where we sit today, it's difficult to tease out which is the most important or whether you have to have that combination and whether that same combination works in all patients. But I don't really care.
I know.
As long as you can get patients, to respond.
And I mean, he's way out ahead of where you might expect tumour recurrence. I think that, I mean, he might expect he's a very fit individual and he might expect to be alive 1 to 2 years after the diagnosis. He's more than 1 year out, but just past the 1 year mark. And I obviously salute his huge grace and courage in being so public about it and giving people insights into what's going on with the therapy and being incredibly transparent. And I know, some of the drug companies were saying, we all want what he got.
Yeah.
But well-designed clinical trials, and I know you'll have to start with phase 1. You can't just go from a case study to a phase 3 trial. But it gives us grounds for optimism that we didn't even have a year ago, because it's moving on the story again. And less optimistic, but also just giving grounds for some level of hope. But I think here it's much more complex. But there has also been 2 recent studies around the use of chimeric antigen receptor T cell therapies. Both in the US, one coming out of Mass Gen in Boston, along with the Harvard Medical School people, and one coming out of Philadelphia, Perelman School of Medicine, and UPenn. And they've both gotten kind of very rapid tumor regressions. But in the Mass Gen study, they were giving the patient's cells engineered to target the epidermal growth factor receptor. And they're also secreting a wild type, an antibody that bound the wild type EGFR protein as well, which is a very interesting little strategy.
And it's not what you would expect, right? I mean, we talked about the blood-brain barrier and how many drugs are excluded from the brain just because they don't make it through the blood-brain barrier. And you think they're small molecules or even slightly larger molecules. But when you've got a cell or when you've got a CAR T cell, you think, how on earth is that going to get through the blood-brain barrier? Well, the blood barrier is vasculated, right? So you'll get blood going through and the cells will go with it. And if they're motivated by whatever they've been engineered with, they'll find the right thing.
But actually, I did take a close look at the paper, though, and it's not an IV infusion. It's actually some sort of intraventricular catheter device.
Oh, right.
That goes into the skull, so it is invasive. And the folks in Perelman, they are using an intrathecally delivered bivalent CAR T cell approach as well. And they're targeting EGFR and interleukin-13 receptor alpha 2. So they treated 6 patients, the Perelman people, and they all had tumor reductions rapidly, some up to 30% or more, but none met the criteria for an objective response, which was over 50% reduction in tumor volume, maintained for at least 4 weeks. also acute neurotoxicity. Yeah, 3 of the 4 patients who had at least 2 months of follow-up had stable disease, which is not bad for a sort of a first-in-human trial of this type. I think the big issue is going to be persistence, isn't it?
Yeah.
Of the
And the
T-cells.
depressing aspect for me is, folks, I don't think we're going to get away without surgery for a long time. Whether it's I don't know, it's neoadjuvant or as in the study you talked about, whether it happens before surgery, it's still going to be nasty.
I think the surgery is I can't say a no-brainer, but it is. I think tumour debulking is essential before you can even think that you will achieve lasting remissions. You really have to, typically, the presentation is usually some form of aberrant neurological activity because of the volume of the tumour. The brain doesn't have a whole lot of wriggle room, as it were, to allow any alterations in volume, any increases certainly.
Yeah.
And so it's usually some sort of aberrant neurological activity.
And this is why most types of brain cancer are diagnosed far too late. It's because, the odd headache here or now that gets worse over a period of 6 months or a year, or behavioural changes that went through a referral to a psychiatrist before they even got to an oncologist or a neuro-oncologist. It's going to take time, particularly in our NHS in the UK. It's going to take decades before you get that. And by that time, as you say, that the bulk of the tumour is much bigger than it was when the patient first presented.
Yeah. There was a real-world study done in the UK recently, and they're trying to present what they think is the reality from their treatment centre. This is somewhere in London. The patients in clinical trials, they claim, tend to be younger and fitter, and the data that they have is very sobering. Glioblastoma, the most common form of malignant brain tumour, 5-year survival rate, they say, is about 5%. 1-year survival rate is 41%. 2-year survival rate is 13%. And the overall survival is 9.2 months. That's median overall survival. So that's a retrospective UK study, just in terms of a large number of patients, just under 500. So that's a sobering.
It is, yeah.
And apparently brain cancer is quite rare. It accounts, from the literature I've read, for 1 to 2% of all cancers. Brain metastases from other forms of cancers are twice as common, actually, as primary brain tumours are. It's a different kettle of fish.
And we talked about my mistake that CAR-Ts can be administered intravenously rather than intrathecally, but then you've got metastatic cancer cells from other cancers, distal parts of the body, making their way into the brain. It's almost like you can't have it one way, but you've got it the other.
Very fair point. And I think you probably get RTs, but you wouldn't making the same passage, but maybe not in the volumes required.
Let's hope so, yeah.
Yeah. And I mean, just to broaden out the study that in terms of what else is in the pipeline, the University of Florida are doing this mRNA vaccine study that's quite interesting. And what's also kind of quite unexpected is that the only other mammalian species that spontaneously gets brain cancer with any kind of level of frequency, apart from ourselves, are dogs. And so they're model species. So in the Florida study, they tested their vaccine first on dogs who developed spontaneous brain cancers who were receiving veterinary therapy. These weren't laboratory animals. These were family pets.
Correct.
So they did the preclinical work there, and then they started going into patients. And it's early days yet in the patients, but they've seen ongoing immune responses in 4 patients thus far. So that's very interesting. What's also very interesting there is that it's not the same type of RNA vaccine in terms of its formulation in a lipid nanoparticle. They're doing this, what they call multilamellar RNA lipid particle aggregates. And they just feel that it actually behaves differently when taken up by the cell.
Yeah.
It's actually recognized by a cytosolic danger sensor called REG1. And that kind of thinks it's seen in RNA virus, and it activates a type 1 interferon response. And that's kind of interesting. Also, there's PARP inhibitors have been tried, and Nerviano Medical Sciences, along with German Merck group from Darmstadt, they're in the clinic with a brain penetrant PARP1 inhibitor, NMS-293. So if they've got the brain penetrant piece right, we've seen with the spectacular results of vorasidenib that you can get activity if you can get across the blood-brain barrier.
And PARP inhibitors are the mainstay of largely ovarian cancer, but also some other cancers as well. But hopefully PARP inhibitors that have been proven to work in some cancers with brain penetrant activity might be a useful step forward.
Yeah, it could be. And then there's an MDH1 inhibitor in development called safusidenib, Anhart Therapeutics, recently acquired by Nuvation Bio. They're on a phase 2 at the moment. In a phase 1 study, they got an overall response rate of 33% in one category of patients who had less aggressive disease and 17% in patients with more aggressive disease. So it's IDH1 mutant grade 2 or 3 glioma. So that's kind of almost on the cusp of glioblastoma if it's grade 3. So slightly different category of patients, I think, from those in the trial of vorasidenib, I think. So there's things happening. Medicinal therapeutics is in there with a very unusual approach. They've got a fusion protein called MDNA55 is the old code name for it. The generic name is bizaxofusp, a fusion protein comprising an engineered interleukin-4 linked by amino acid linker to a Pseudomonas aeruginosa exotoxin. And that's in unresectable recurrent glioblastoma. And they're claiming 13.5 months median overall survival from a phase 2b trial versus 7.2 months from matched external controls, individuals who weren't on that trial but matched the patient characteristics. Very interesting approach. It's just a single dose delivered in tiny amounts, because the idea of putting a bacterial toxin in your brain sounds a little bit counterintuitive, to put it very mildly. Yeah, and another slightly unusual interleukin-based approach is NeoImmuneTech. They have a product called NT17 or efineptakin alpha, an engineered interleukin-7, which has a longer half-life. It supposedly promotes the expansion of naive memory CD4 and CD8 T cells. And they're in a phase 2 combination study with pembrolizumab, the PD-1 inhibitor, in recurrent glioblastoma. So that's interesting. I mean, you can see the rationale there if it works, but I haven't seen any data. They're trying that in a whole bunch of different cancers. So there is a pipeline, and there's other things. Avita Biomics are doing botanicals based on curcumin, but basically that's not very brain penetrant. So they have a bunch of other ones from this extract, I guess, which offers more activity. So they're doing a phase 1-2 combination study with bevacizumab. You really don't know whether it's going to kind of put it for such a difficult...
As you've talked about, Cormac, lots of things are finally happening in wildly different mechanisms of action in brain cancer, where for years we never had it. So, I think the take-home message is for the patients and the families of the patients that, as you said, these are rare cancers.
Really?
And so perhaps the normal rules, I'm sure the normal rules don't apply. So for patients and their families, if, get them to talk to their oncologist about enrolling in a clinical trial, because these are rare cancers. So the more data that can be generated, as we've seen some fantastic data, the better it will be for the future or future patients.
Yeah. And so I think the MEK work for better brain penetration is very important. The ongoing work trying to figure out how the tumor microenvironment, the immunological status of the tumor microenvironment, that's very important. So there's things happening, as is always the case for patients. It all happens too slowly, but there is hope. Yeah. And your son's friend, I didn't quite catch his name at the start.
Jacob Whitehead.
Yeah, well, we remember Jacob Whitehead on this episode, and I'd also like to remember a cousin of mine, Nick Bowen, as well, who passed from disease last year. Yeah. So I will hope for patients currently living with the condition that they at least get some level of therapy that will give them some additional quality life extension. It's too early at this stage to start talking about cures, but that ultimately, of course, is what the objective is.
It makes you feel for those patients who have, like Jacob, who've got the diagnosis and have succumbed to that disease in the past, right? Because you think back to the early days of HIV when people were dropping like flies. So and then suddenly we have small molecule oral drugs that allow them to live normal lives. Okay, brain cancer is more difficult than HIV, but everything is in the future, isn't it?
Yeah, and there is work going on because we've seen some glimmers of success that encourages other people to mobilize resources and get active, and that's what leads to possibilities. So thanks for listening, folks.
Thanks, everyone. Bye.